I would say that I've always been interested in science as long as I can remember. I mean, my father was well, he was an engineer, but he worked at Bell Labs on trying to understand how to synthesize speech, and so it was kind of science. I guess in terms of things that I did, I can remember my grandparents lived in the rural Nebraska. In Paleozoic times, there was a sea that covered the entire middle of the United States, and so there's all these limestone deposits that are full of fossils. So I definitely remember going out as a kid in the summers when we'd go out to visit my grandparents and hunting for fossils. So that was kind of like science, I guess, an act of discovering something new that was quite fun. Yeah, my name is Bill Schafer. I'm a group leader in the neurobiology division at the LMB. My career, well, let's see. So I was born in the United States, and let's see, as an undergraduate, I was at Harvard, and I did undergraduate research in the lab of Richard Lozick, studying bacillus gene regulation. And then I did my PhD at the University of California in Berkeley. And there I worked on yeast. I studied sort of lipid modification of proteins, and I was in the lab of Jasper Rhine. And then as a postdoc, I switched to studying C. elegans, and so I went to the lab of Cynthia Kenyon at UCSF, and that's when I started working on trying to understand behavior and the nervous system. So that was really my first introduction to neurobiology. I had never studied neurobiology as a student. I kind of learned about neurobiology by reading books on the subway, commuting between Berkeley and San Francisco. But it's kind of easy to learn. I mean, the studying things in worms was fairly similar to studying them in yeast, in that it was like a simple microscopic organism that had good genetics. And so conceptually the approach was similar, even though I was studying a different problem. And so then I had my first independent position as a assistant professor at UCST, University of California, San Diego. And I was there for, well, so I had been there for 10 years, and then after I got tenure, I did a sabbatical here at the LMB in 2004, and I liked it here. And so before I left, I asked Hugh Pelham, who was he was then the head of cell biology and was going to be the director, if there were any jobs here. And I guess there were, and so I had to go back for a year, or they would have made me give back my year's sabbatical salary. But then I moved my lab here, and so I've been here ever since 2006, I guess. So yes, that's my that's my story, I guess. My current research, well, so it's hard to summarize everything that we're doing. I mean, most of my work since being since being a postdoc has been on C. elegans, so studying various aspects of how the nervous system works using C. elegans as an experimental organism. And so the reason for that is that because of work done here by John White, we know the entire structure of the nervous system. So we know the identities of all the neurons, and we know how they're connected, and it's easy to do genetics, and it's easy to do behavioral assays. And through work that we contributed to, it's possible to use optical techniques to record neural activity while the worms are doing behavior. So it's possible to try to break down behaviors into sort of their component parts and try to understand how an entire nervous system works at the molecular and cellular level. So we've studied a variety of problems along those lines over the years. We've studied how sensory pathways work. We've studied how chemicals called neuromodulators control behavioral states. And most recently, we've focused on a particular group of these neuromodulators called neuropeptides, which are the most molecularly complex group of chemical signals in the nervous system. So even C. elegans, which has only 302 neurons, has hundreds of different individual neuropeptides that each bind to specific receptors. And so there's probably 150 different neuropeptide binding receptors, even in a nervous system that only has 300 neurons. And so, one of the things we've been interested in is in trying to understand how these biochemically complex networks sort of relate to the synaptic sort of wiring diagrams of the nervous system to make the whole thing work. So that's sort of one aspect of things, is we're sort of trying to understand how these so-called wireless signaling networks of neuropeptides and other neuromodulators are organized and how they control brain activity and behavior. And then we've sort of gotten interested in recent years in sort of expanding out of C. elegans. So this, coincidentally enough, is a picture of an octopus brain. So I had a postdoc who joined the lab about 4 years ago, Amy Courtney, who was interested in looking at some of these questions that we've been studying in other animals. Specifically, this was not neuropeptides, but looking at receptors for other types of chemical signals, so fast classical neurotransmitters. And I have a sort of a sidelight at the university in Leuven in Belgium, and one of my colleagues there studies octopus. And so we sort of thought it would be interesting to try to look at some of these gene families that we've been studying in C. elegans in the octopus. And so, it's turned out to be very interesting, because it turns out that at the chemical level, the octopus brain works quite differently from the sort of mammalian brains that we're familiar with. And so we've been able to kind of apply some of the approaches that we've worked out in C. elegans to this animal with a much more complex brain that's actually much harder to work with in a lot of ways, because that doesn't have the genetics, and it's not as easy to grow them in the they basically have to capture these animals at sea, and then we work on the larvae that hatch in their millions when these animals. So we can sort of use these larvae to apply C. elegans-like approaches to try to understand their brains. So we also have some sort of non-worm projects ongoing these days, and so that's one of them. The sort of bread and butter techniques that we've used, for 20, 30 almost years. I mean, so C. elegans the nematode was sort of picked as an experimental organism because you can do genetics, so you can basically do molecular genetics and crosses at the bench like you were working with bacteria or yeast. So you basically set up crosses, you make mutants, you try to figure out what the gene that's defective in the mutant is, and then you study its phenotype. And so lots of people day to day, spend their time doing these worm crosses. So they'll be sitting at the bench and they'll be picking worms, they'll be mating different strains, and then once they get the strain they're interested in, they can do various assays to see what's wrong. And so in terms of the assays, one of the things that I have been interested in from the beginning is trying to use automated machine vision microscopy to try to quantify behavioral phenotypes that we see in worms. And so, you can make a little automated microscope that will make long movies of worms over time, and then you can take these videos and try to extract features of the behavior that are relevant to how the brain works. And so there's lots of people that spend their time doing these worm tracking experiments as well. And then sort of the third thing that again, we were sort of one of the first people to do is to use genetically encoded fluorescent probes to try to visualize neural activity while worms are doing behavior. So we can express modified fluorescent proteins that are sensitive to neural activity, and then we can look and see how particular identified neurons in circuits respond to a particular stimulus. So that's those are sort of the worm experiments. But then we've also gotten recently into doing more electrophysiological experiments, and this wasn't really a technique that, it was definitely not a technique that we invented, but you can basically just take worm genes and produce their RNA and inject it into frog eggs. And then from these frog eggs, you can stick electrodes in to the oocyte, and you can record the activity of the worm. So basically, the gene that you inject is the gene that encodes an ion channel or a receptor or something like that from a worm, and then this frog egg will produce that protein on its surface. And then you can use this sort of so-called two-electrode voltage clamp technique to record the activity of the channel or receptor from the worm in a exogenous environment. So you can look in isolation at a particular channel or receptor. And we have automated two-electrode voltage clamp rigs, and so we can, we can do lots of experiments in a day and look at lots of aspects of a particular channel. And this is the technique that was sort of our way into the octopus, because of course if you can inject worm genes into a frog egg, you can also inject octopus genes. And so that was where we sort of got into this idea of exploring the properties of octopus neurotransmitter receptors. And then once we found interesting features of these receptors, then we could go back to the octopus brain and see where these were found in the brain, and ultimately, how they affect the activity of brain slices and so forth. So that's sort of the other thing, is that we have these sort of in vitro techniques where we use frog eggs to produce worm or octopus or other animals' ion channels, and then look at their properties. Yeah. As I was saying, when I was a graduate student, I worked on yeast cell biology. But I also really enjoyed sort of intellectually the process of using genetics to try to understand the problem. At that time, I was not somebody who was particularly handy at the bench, but I could always figure out how to do a genetic cross and try to interpret it. And so I was interested in the brain, and I thought it would be interesting to try to study how the brain worked. But the only way that I could understand how I could attack a problem like that was using genetics. So I just thought, well, I guess if I want to use genetics to study the brain, I could either work in C. elegans or I could work on flies. And I don't know, there were then I don't know why I decided worms was better. Maybe I can't remember, it was too long ago. but that was really why. And at the time, Cynthia Kenyon didn't really work on neuroscience. She worked on developmental biology, and then later she worked on aging, as many people probably know. That's what she's kind of famous for. But she was interested in having me work in her lab on a neuroscience project. I don't know how I convinced her to do that, but she was very supportive, and it was great. So yeah, the thing that I think is most exciting now, and is still, I think, it's probably is also useful in the scope of things. But I think that this question about how neuropeptides act in the brain is a really fundamental question. So, as I said before, in people are very interested now in mapping the connectomes of brains. So people are trying to understand how all the wiring between all the neurons in the human brain, and also and before they do it for humans, the brains of smaller animals, are organized with the idea that that's going to tell us how our brain works, and that maybe if we knew how all the wires were put together, we would understand enough about our brain that we could make a model of how it worked, that we could understand it in terms of its parts. But we've had that kind of map for C. elegans since 1986, and it's only 302 neurons. And clearly that's not enough for the logic of how the brain works to just fall out in an obvious way. And so there's lots of explanations for why that might be, but one explanation is that the communication in the brain is not limited to communication between neurons that are wired together. So a lot of these neuropeptides allow neurons that aren't connected by synapses to communicate with each other. And we know that this communication is really important because lots of the drugs that we know of that affect our behavior, like Ozempic or opioids or things like that, affect these neuropeptide pathways that even in our brain are acting through these wireless communication mechanisms. And so to try to understand how these networks are organized and how they work is really important to understanding how brains work. And through actually the work of a really good student who's about to defend her thesis, Lydia Ripoll Sanchez, we've actually been able to make a map that covers neuropeptide signaling across the entire C. elegans nervous system. So we can basically map the connections between all the neurons individually that are mediated by neuropeptide signaling. But obviously we'd like to be able to extend the insights that we get from C. elegans to bigger brains. And so one of the things we'd like to do is to look at octopus because of course octopus also has lots of neuropeptides and it has a much more complex brain. And so, that's one place that we'd like to sort of scale up and see if we can look at what those networks look like. But obviously we're also interested in collaborating with people who are studying mammalian brains to try to see how the features of smaller neuropeptide networks from animals like C. elegans, how they scale up into mammalian and human brains. But it's a big job, and it's it's not, that's not going to be done right away. But I think that the work that we're doing in C. elegans hopefully will reveal basic principles for how these signaling networks work and how they interact with wired synaptic networks that will be informative to people that are trying to study these processes in bigger brains. When I was first setting up my lab, I was mostly interested in behavior, so I was trying to set up these worm tracking experiments and so forth. But a student came into my lab and said, "Oh, there are these new genetically encoded calcium indicators that have just been developed, and I think if we use these in C. elegans, we could understand how the entire brain works and make a physical model of it in my thesis project." And I thought, well, that's wildly ambitious, but if it works at all, then at least we'll have a way to actually see what the activity of neurons is using our behaviors, and it will be incredibly useful. And so I said, "Oh yeah, sure, so why don't you give it a try?" And it turned out that it was actually not that easy. A lot of people were trying to use these indicators at the time without success. And this guy, whose name was Rex Kerr, was a very talented student. He knew how to do genetics, but also how to do computer programming and how to do optics. He had also a undergraduate degree in physics as well as biology. And so, he was able to solve this problem, and this actually opened up, all kinds of exciting avenues. My lab sort of dined off this result for a number of years, and eventually people, without not too much longer than that, people started using these in flies, and then later in zebrafish, and then in mammals, and now people use them all the time, and probably most people don't know that for a couple of years we were the only people doing this. But it was sort of it definitely changed the direction of the lab. And this is a case where, there was completely the idea of a student. It wasn't really my idea to try these out. So this is a thing that I think is fun about having students in the lab, is that a lot of times they come up with ideas that I wouldn't have had otherwise, and I try to help them realize their goals. But it's nice when students and postdocs ideas also sort of that it becomes sort of a collaboration between me and students and postdocs in the lab to sort of make new discoveries. Well, if we're talking about experiments that were at first glance unsuccessful, or at least inexplicable, that turned out to lead to important progress, one that I can think of involved another student named Katie Kint. And so she was working on neural circuits in the worm that were involved with the sense of touch. So if you touch a worm on its nose, it will crawl backwards to try to escape from what it thinks is a predator or something at least bad. And so she was studying a mutant that was in a gene that turned out to be in an ion channel that responds to mechanical stimuli that's important for the sense of touch. And she found that these mutants, indeed, when you knock out this gene, they were defective in this escape response to nose touch. So when you do genetics in worms, the next thing that you want to do after you see a phenotype, or at least if you're doing neural genetics, is you want to try to rescue the mutant phenotype, in particular neurons, and that tells you which neurons it tells you in which neurons this gene has to function in order to have normal behavior. And so she started doing these rescue experiments, and there was one neuron that everybody knew was the most important neuron for this escape behavior, because if you kill it with a laser, the worms don't do the behavior. And so that neuron did express this gene. She knew that from the reporters. But when she did the rescue experiments, it didn't rescue. On the other hand, when she tried to put the wild type gene back in other neurons, she did see rescue. But it was really sort of weird, because those neurons were supposed to be involved with other behaviors. They were thought to be mechanosensory, but they weren't supposed to be important for this escape behavior. And so it was really very puzzling, and we didn't really quite know how to understand this result. And for a long time, we reported it, and we said, well, this is a mechanosensory thing, but we don't really understand why these other neurons are important for this escape behavior. And it was only a few years later, when I had another student in the lab, that we came up with the answer, and that was that there was another circuit where these supposedly unimportant neurons were connected by something called gap junctions, which are electrical connections between neurons with the neurons that were in this escape circuit. And it turned out that there was this mechanism of sort of coincidence detection, where you had to activate multiple neurons that were connected by gap junctions into this escape circuit, and that would give you this behavior. And for reasons that are kind of complicated, the cell ablation experiments where you killed those neurons with a laser didn't give the same result as changing the activity of those neurons with this mutation. And so this was a case where, something that sort of gave a weird result that maybe we would have liked to sweep under the table actually led to an unexpected insight into how these circuits worked. I'm not sure that I would say that I have a scientific hero, but I would say that I'm very grateful to all the mentors that I've had over the years, all of whom have hopefully taught me how to be a scientist and do my job. I mean, when I was an undergraduate and worked for Rich Lozic, you know, maybe it's sort of silly because most people when they're in college, you know, they are more interested in, I don't know, screwing around and going to college, but I are going to parties. But I just loved going into the lab and hanging out with graduate students, and I just thought that, I don't know, I sort of, it was just sort of a fun environment to hang out with and talk about science. And, you know, he really had, I don't know, it was a very appealing environment to just come and do science and talk to people. And, you know, it was very stimulating. And similarly, as a graduate student, I thought that, you know, Jasper Ryan was a super supportive mentor. I was not, I mean, I always was ambitious, and I maybe was able to think about science, but I was not maybe a natural bench scientist, and he always had confidence in me that I would be able to do good things, and it was really important to me at that time. And similarly, Cynthia Kenyon was also great. I mean, I always, even, you know, from the minute that I came into the lab, I always had complete independence to sort of do the things that I wanted to do and come up with ideas. And she basically never told me what to do, but she always had good insights and good advice for what experiments to do next, and was always there to talk to me when I had questions and when I wanted to talk about results. And so I think that, you know, all of the people that whose labs I worked with as a student and postdoc all had this idea of giving people a lot of freedom, but also being supportive, and, you know, giving both, you know, personal as well as scientific support when, you know, things were going wrong or people had questions, but never tried to micromanage or tell me what to do. And that's something I've tried to do in my own group, and hopefully I've succeeded to some extent, I don't know. But certainly all three of those people have been real role models in terms of how to run a group and how to be a scientific mentor to students and postdocs. I mean, when I came here on sabbatical, I was a newly tenured professor at UCSD. And, you know, I was probably, I think I had just turned 40. And, you know, already, you know, I had lots of teaching large undergraduate courses of 400 students, and I was spending time writing grant proposals all the time, and I was having to serve on committees, and, you know, I could already sort of see that, you know, the things that I had enjoyed as an assistant professor of sort of interacting directly with students and being actively involved with the research was getting more and more difficult. And then I came to the LMB, and, you know, basically when I was here, I was just messing around at the bench. But I could see that the group leaders here, you know, they had smaller groups, it was true, but also that it was easier for them to be directly involved with research. Some of them were actually still doing experiments, but even the ones that weren't, they had so many fewer constraints on their time that they actually were playing an active role in guiding the research, writing the papers, thinking about experimental questions. And I thought, this seems ideal. It's almost like I could be an assistant professor for my entire career. And so that's what motivated me to try to move my lab here. And I would have to say that it has more or less turned out to be true, that I can't say that I do a lot of bench work myself. I basically don't do anything except maybe look at fluorescent images of worms and try to identify cells for people. But I would have to say that I've always felt like I've been able to stay involved with the research in a way that would have been very hard in an American university, for example, or maybe a British university too. I don't know. I've never worked in one. But yeah. I think it's hard to answer the question of what skill in the singular it takes to be a good scientist, because I think different scientists have different strengths and there's different ways to be a good scientist. And I certainly don't think that I have all of the qualities of a good or great scientist. And you see that also when people come through the lab. Everybody has strengths and weaknesses. And I think the key is to figure out what your strengths are and to take advantage of those in your experiments and in your scientific work in general. So, and I guess you can say that as a group leader, I guess maybe part of my responsibility is to figure out what the people in the lab's strengths are and to help them improve the things that are not their strengths, but also to take advantage of the things that already are their strengths and to allow them to be successful. So, yeah. If you think intellectually about science, I think that there's kind of two sides. There's a side of imagination, so trying to figure out, what, both in terms of thinking up interesting questions to investigate, but also thinking of interesting hypotheses about how things might work. But then the flip side of that is being critical because, it's very easy to think you know how things might work, and then you do an experiment, and maybe the results don't turn out exactly like you thought they would. And so I think you also want to have someone who can apply a critical eye to things and adjust their hypotheses based on the evidence rather than trying to have it fit your preconceptions. And so I think that there are certain people that tend toward that are really good about the imaginative side and have a harder time being critical. And I think there are people that are too critical and have a hard time being imaginative. And I think it's, in some ways, that you can deal with that in the lab by having a mix of people with different intellectual approaches to things. But also, I think as an individual, it's good to try to work on to work to counteract your natural tendencies to excess in either the imagination or the critical thinking. But I would say there are two things I like to do away from science. So one of them is sports. So I, for a long time, I still kind of do it, but for a long time I was a rower in one of the clubs in Cambridge, or a couple of the clubs in Cambridge, and I used to really like to do rowing races. And then through various injury issues, I had to sort of retire from competitive rowing, but now I've sort of taken up running again, which I've always done running since I was in high school. But in my guise as a professor in Leuven, I've joined a running club there, and so I've started running in races again. So I quite enjoy that. And then the other thing I like to do is read. So I like to read books. And the other thing, this is again something that I've picked up from working in Belgium. One of the things that I had to do was I had to, in order to have a job at a Flemish university, you have to learn Dutch. And even though I had a part-time position and I probably could have gotten away with it, when I started working there was during the pandemic, and I didn't have a lot of things to do, so I sort of got fairly seriously into learning Dutch, and so now I actually mostly read books in Dutch. So my two hobbies are long distance running and reading books in Dutch. I would say that actually I can't really complain too much about my scientific career. I I think that, you know, I'm quite an old man these days, and so I've probably have more of my career behind me than I have ahead of me. But I would say that I've been lucky to be able to work with lots of talented people and see them go on to start their own labs and be successful, and that has been great, and it's been maybe beyond my expectations. I've been able to make a few significant discoveries, which I can take pleasure in, and I'm still at this late stage of my career doing new things and going in new directions, and I've been able to change direction multiple times over the course of my career, which has been good. So yeah, I honestly have nothing to complain about my career, except maybe a few failed grant proposals. But otherwise, I think it's been good. Well, I would say one thing is that I would definitely advise or encourage is that if you want to do science and you want to, especially if you want to be, you know, run your own group and be an independent scientist, I think it's very important to, this is going to sound trite, but is to not get discouraged or not to give up, because the process of doing science, there's small disappointments every time an experiment doesn't work, and then there's big disappointments when, you know, a paper that you submit doesn't get good reviews or your grant application gets rejected. And I think it's very easy to get discouraged and give up. And I think that it's maybe not the single thing. Obviously, there's some requirement for being able to think and do experiments and so forth. But I think that the thing that separates the people that go on in science from those that don't, a lot of it comes down to how you deal with rejection and frustration and failure. And I think that the main thing that I would advise people is not to get discouraged and not to give up, and to have confidence in your ideas and your abilities. Yeah. When I was thinking about where to do a postdoc, I had sort of briefly considered the idea of working on zebrafish, and so I had contemplated the idea of going to work with Janni Nusslein-Volhard in Tübingen, and I didn't do that for various reasons, personal reasons and otherwise, but in retrospect, that would have been kind of cool, because she's sort of, I said that I didn't have scientific heroes, but she's kind of one of my scientific heroes. And I think at that time, in the early days of the zebrafish, you know, would have been a quite exciting time. And so that might have been, you know, wouldn't say it was a regret, because I had a great time as a postdoc with Cynthia Kenyon, but it might also have been fun to be in those early zebrafish days. So, yeah.